Welcome everyone to the AIRR-C seminar series of September 2024. As always, we have two very exciting speakers, one more established speaker and one more junior speaker. And we're going to start off with Sol Efroni, who briefly to introduce Sol, he did his Ph.D. at the Weizmann Institute and then his postdoc training at the age to work on personalized cancer genomics. And then he returned in 2009 to Bar-Ilan University, where he's now focusing on T-cell receptor repertoires, which is also the focus of this talk in conjunction with language models. Very much looking forward to your talk. So, and afterwards we have Giulio Isacchini, who will talk to us about generative modeling of air repertoires with Sonia. But first, Sol.
Hello, everyone. Thank you, Victor, for the introduction. Let me share so I'm trying to see if I can share. Let's see if this works. Working, Victor?
Looks great. Looks great. Yeah.
Okay. Thank you. So I'm going to talk about this long title. As Victor said, this is about T-cell receptors and language models. What you see here is actually Bar-Ilan University, and this photo has been taken by my friend Alon Kongrin. And as you can see, Bar-Ilan University were kind enough to build this administration building in the shape of a T-cell receptor. Um, and so as Victor said, I came to Bar-Ilan about 15 years ago, and we worked mostly on personalized cancer genomics. I did my postdoc at the NCI, working on the cancer genome atlas, but then more and more we worked towards T-cell repertoires. Actually, the first grant application I ever made was about the T-cell repertoire in zebrafish. But around 2016, 17, we moved almost completely into T-cell repertoires, and this is what almost everyone does in my lab. And the title of this talk is as the title of this. I'm going to talk almost completely about one paper. This paper that was published a few months ago. That has the same title. It's about T-cell receptors and their properties as those are seen through self-supervision. And these are the people who worked on this, mostly Romy, and I'll show what Sarit and Avital did a little later, and Alona does everything, as you will also see. So just to set the stage as to when all this was happening, this is when we started. So this is COVID, and this is a nice timeline as to when we started this. This is about the time we started this. And you can imagine everyone working from home, which actually didn't change much since then, but it was kind of new at the time. And everything was happening over Zoom, and everything was happening through Slack. And this is just to put you in the state of mind that we were in. And Romy, who's a really good student and started not long before all that, came to me and told me she started working on single cell time, on gene expression inference and things like that. And she came to me and told me that what she actually wanted to work on was transformers and large language models, and she wanted to see how those could be connected to the T-cell repertoire. And to be honest, I did not think anything would come out of it. I said yes for two reasons. One of them is that she's really smart, and she kind of knows what she's doing, and I didn't want to interfere with her wishes to work on something like that. And the other reason is that I sometimes am too nice, but in this case it turned out nicely. But I did not think that this would come up to anything at the time. I have to admit. So she'll So she said she wanted to look at it, and I wanted her to continue what most of the lab has been working on and is still working on, which is the use of the T cell repertoire for mostly as a biomarker. This is what we're focused at. We are trying to get as many clinical samples as we can and see if we can make something out of the T cell repertoire, out of those samples and out of their repertoire. So I wanted her to work on this, but I agreed to working on this other thing because it also sounded really interesting. When we started working on transformers, and I'll explain more about this. So I told you this is the peak of COVID at the time, and this is something that Romy sent over Slack April 14th, and you'll see why I'm giving you these dates in a minute. But what you see here is that the model actually, the loss is going down on a model. I'm going to explain the model in details, but what you see here is that it started working. So not long after she started working on this, the model says that the loss is going down, which means that the model is learning something, which is exactly what I did not expect. So I was totally wrong. And Romy was able to train a lot of sequences at the time and more of them later. But it seemed as if the model was learning something, which was really surprising. And again, this is April 14th. And so things just started then. And the first thing Romy sent me after showing me this loss was a few days later. And what you see here, these elongated clusters, this is a UMAP of the, again, I'm going to explain in detail, but this is a UMAP of the 768 dimensions that are the model. This is a UMAP in 2 dimensions. What you see here, I'm going to show you a lot of UMAPs. Most of them are not going to be screenshots. But what you see here are T cell receptors. Every dot is a T cell receptor, or I should not say a T cell receptor. Every dot is a CDR3 sequence. Those are all CDR3 sequences. But what you see that are in blue are public sequences. And you immediately see that something is going on here. First, there are clusters. There is a structure to this. That's one thing, but another thing is that the public sequences clustered together, they are together. No matter what you see here, you see that the blue dots are together. They are at the tips of those elongated clusters, but they are together, which was absolutely surprising for me. So that was the very beginning of all this, and it's kind of one of the first images that we saw. So that was April 18th, and then a few weeks later, this happened. This Systems Biology and Immunology Symposium in honor of the late and great Neil Friedman. So yes, I showed a bunch of these things at the meeting, and again, this was during COVID. I think we were with masks still. I don't remember the details. I actually don't remember everything I talked about, but I do remember showing this because the minute I showed this, Thierry Morin said, "Okay, so these must be J genes." That was something that we needed to check. And so again, that was 18th. Sorry, that was May 2nd. And then a few months later, because Romy had a baby, a really cute baby, that is now 2 years old. So a few months later, Romy came back with this. So what you see here is another one of those UMAPs. As I said, I'm going to show you a lot of them. But now the dots are colored according to their J genes. And it becomes immediately obvious that these clusters are, as Thierry said, those are J genes. So that was cool. Later, we saw that it's that the J gene clustering and the J gene classification are kind of simpler tasks for this model, but at the time, this was just a really cool thing to see. So going to explain about the T-cell receptor to this audience. What I am going to say is that the T-cell receptor is a sequence. And by saying sequence, it means that the letters, the amino acid representation that is letters, comes one after the other. And if I take this sequence, and if I take these amino acids, if I take these letters and scramble them, I'm going to get either a meaningless thing or something very different from what we see here. And this sequence and these and then thinking of amino acids as words makes this immediately available for analysis with the tools of language. And obviously, we are not the first. So as I was saying, a lot of work on language models and proteins and protein understanding, and as you can see, a lot of them, not a lot of them, but at the time, some of them were using BERT, protein BERT, DNA BERT, and we were also using BERT. Now there's a lot of transformers or language models to choose from. Even then, there were definitely more than one, but BERT was kind of perfect. It was definitely the first thing we needed to try. And if you don't know what BERT does, this is what it does in the language world. So there are sentences. When you train, when you learn, you mask. That's the self-supervision. You mask some of the words, and you encode the words using token embeddings, which with words there are a lot of word embeddings. So you can put the word embedding, and you also put positional embedding, because as I said, in protein and is obvious in sentences, the position is of critical importance to the understanding of the sentence. So we have position embedding, we have token embeddings, and this transformer produces an embedding for this basically sentence. Now, BERT is an encoder. The decoder is different in BERT than the original transformer paper, because the decoder is simply a classification task. So the BERT will choose this specific word out of an option of all the words that could be here, and it's just a classifier, and it works really well for BERT. Works even better for current day models. But Romy decided to try BERT, and it worked really well. So as I said, this is the representation in a sentence world. And this would be the same thing for amino acid sequences. This is just these are just 6 amino acids, but you can see how it would work when we have longer sequences. So we trained the model exactly as we would train the model with the sentences. We provide it with a lot of sequences. This is what it looked like, the training itself. So we based it on the BERT architecture. We used a lot of sequences. We used more, we used less. It doesn't really matter. It stayed very stable. The model was taken from Hugging Face, and then this model, when we used it for T-cell receptors, we just named it after. So there is this tradition of naming. There used to be this tradition of naming transformers based on Sesame Street characters, like BERT, and that's another Sesame Street character. It's called Count von Count. Here's a rare footage of the count with BERT, and this is what the whole process looks like. So we each sample, and we have 2,400 samples that have been collected by Sarit a while ago. So we have a lot of them. So we took all these samples and into this large table. As you can see, the rows are CDR3 sequences, and the columns are samples. So what we have once we have this large table are two things. First of all, we just have the unique sequences. So we have 92 million. And sorry, I didn't mention this. These are still beta sequences. I will get to alpha sequences in a minute. So these are beta sequences. So we have 92 million CDR3 sequences. By the way, you can see my pointer, right? You can see the mouse.
Yes.
Thanks, Google. So we have 92 million sequences. That's 91, but it should be 92. First time I noticed this. So we have 92 million sequences, and that's one thing we have from this large table. We have the list of long sequences, and we can take this long list and use it to train the model. As I just said, we do it in this case. We did it in Google Cloud. We take some of them or all of them, depending on the computational power that we have, and we train it, as I said, using self-supervision. We mask some of the sequences, we use it to train the CV model. So we have this CVC model, and now these sequences are no longer represented as CDR3 sequences. They're no longer represented as amino acids. They are vectors in 768 dimensions. That's what the model does to them. It takes a CDR3 sequence, and that CDR3 sequence becomes a 768 dimensions vector. And now in 768 dimensions, it's kind of difficult to see anything. So we do a dimensionality reduction. We do UMAP. And in UMAP, you can see a lot of things, and I'm going to show you some of them. And you can make you can come to a decision as to what works and what doesn't according to this representation in 2 dimensions. Now, I said one thing we have is this long list. The other thing we have is knowledge about the public status of these sequences. So if a sequence is in more than one sample, we can call it public. We cannot only call it public, we can also count the number of times that this sequence appears in samples, and we can count its publicity. So public is yes or no. Publicity is a number between 1 and 2,500, 2,400, depending on the number of samples that we have. And once we have that tag, that status, we can do supervised learning. So we can take this embedding and use different machine learning algorithms to see if the embedding provides us with the status of the sequence. And once we do that once we train the machine learning model with enough sequences, we can make predictions. Okay, so we did that to 100,000 sequences. Now we can do it to the 100,001 and ask if that sequence is public or private. And in that way, we can not only see kind of a representation in UMAP, but we can also quantify how well this hyperdimension actually separates the sequences into the tags that we provide it with. So we did all that, and as I said, we started with one quality that we could measure well, and that one quality was the status of public or private of a sequence. And for me, I have to say that I did not think that this, I saw it at the very beginning. As I said, we saw those blue dots at the very beginning that showed us that CDR3 sequences that belong to public sequences are somewhat separated from the others. But I couldn't think of a really good reason for that to happen. But the fact that CDR3 sequences contain within themselves this property, this knowledge of whether or not they're public was super interesting to me. So this is the first thing we did. We took all those sequences, all the sequences that we have here. We just projected them into a UMAP, and then we colored them according to their public and private status. And this is what happened. It's kind of similar, only kind of a little better than what we saw at the beginning. So what we have here again is a UMAP. Every dot is a TCR sequence. The private ones are colored in yellow, and the public ones are colored in blue. And again, you can see now maybe better there. You'll see it again and again and again, but you can definitely see that the public sequences have their own little places at the edges of these clusters. That's one thing. and you can actually see those clusters. You can see them very well. Now, the fact, so again, this is a projection from hyperdimension. And in such a projection, you don't really know that, for example, these blue dots here are far from these blue dots. You do know that they are close to one another, but you do not know if there is another way to represent these 768 dimensions in which the blue dots, all the blue dots will be on one side of a hyperplane, and the yellow dots will be on another side of the hyperplane. This is something that is yet to be seen. So that is one thing we saw. I'll get back to this in a minute. So this is one thing we saw. Another thing we saw is that you can actually see some gradient in that hyperspace, in that multidimensional space, some sort of gradient of sequence length, which is another interesting phenomenon that just pops out of these images. Another thing that I did not expect and definitely happens. Sequence length is a direction in this multidimensional space. It's not clear what direction it is. Another thing we saw was a convergent recombination. So if you add another tag to each of those sequences, just count the number of DNA sequences that would code for the same T-cell receptor sequence, you see that those provide another gradient that we could look at. That you see, those are the red ones are convergent recombination at the level of 0 to 100. And those are at the very top. So those are a lot of convergent recombinations. They have their own little place within this multi-dimension, which again, simply surprising. But again, it's only a thing that that you could see, that you could look at, not necessarily something you can quantify at the moment. But convergent recombination was another gradient to work along. Now, so we had that, but and that was the beginning. For the first few months, this is what we saw. But then we said, what if we had more data about these cells? So I didn't say cells until now. I only said DDR3 sequences. But we can look at cells. We can look at specific T cells, and we can look at them because we have data, because there is public data that we could look and collect that tells us about single cell TCR, single T cells. So when you do TCR sequencing, you can look at bulk, and it looks like this. You would know that this is a TCR, and you would know what it is, but you cannot pair it with anything. Same for this one. You know it's a TCR, you don't know what to pair it with. And in single cell work, this all gets organized really well, and you know that these are pairs, this is a pair, this is a pair, and so on. But the other thing that you have as soon as you have single cell data is you have a gene expression abundance. So when you do single, and I'm talking now only about, not only, but I'm talking about 10x data. And there is a lot of 10x data out there. So when you have a T cell, you not only know that the pairs of alpha and beta, you also know gene abundance. And from that gene abundance, you can say a few things about that T cell. So we took a lot of data. Avital collected a lot of cells, more than 2 million cells. And basically, once we had those 2 million cells, we did the same thing. This is single cell CDC. So Romy built another transformer that is based on single cell data. And it's the same concept, basically going through the same steps, but this time the alpha and beta are the sequence with a token between them that allows us to link them. So when you do this self-supervised learning, and BERT is bidirectional. It knows it either came from here or it came from here, and it can look at the token from both sides. So when you do self-supervised training on the transformer, you get, it's a BERT, so you get 768 dimensions, but this time those sequences are alphas and betas. And since you know the alpha beta, and gene expression, you can look at the at a list of other things. We looked at mate because you can know from these data which cells are mate cells and which cells are not mate cells. So you have, for example, you have a bunch of other stuff, but for example, you would have this. You would have a tag that would tell you this cell is a mate cell, this cell is not a mate cell. And for example, you can do um, classification over these data as well. Um, so actually, I think I went over these for you. You know what these are, and let me just skip this one as well. Okay, so now I said we had a few tags that we could look at. So let's look at two examples of tags that we had and we could use to check if to check for the meaning of this hyperdimension, or for the meaning of the vector representation in 768 dimensions of the CDR3 sequences. So if that hyperspace provides us or contains information about um, public and private T cells, we could devise a classification task, a binary classification task that would let us know how well this works. And this is what we did. We took these embeddings and we went through the task of binary classification to tell public sequences, or to see if we can tell public sequences apart from public sequences from private sequences. So that was one task. The other, as I said, was looking at mate cells, and you can see. So as I said, now we have gene expression for mate cells, so we can see if we can tag them in that way. And also we have alpha and beta pairing. Usually we don't have alpha and beta pairing. We would have alphas or betas, so we can, which I think is more interesting in this case, see how the beta behaves when we have alpha. And so we can play along this gradient of do we know what the tag of that cell is, and can are the other factors that we have, are they able to let us know anything about this about the tag that we are after? So we did the same thing for these are the single cell CDC embeddings, and this is gene expression data. And again, we went through binary classification to look at mate cells and non-mate cells. And the last thing we looked at was if we can do a multi-class classification. And in this case, this is just the J genes. And we wanted to see if those embeddings provide us with enough information. And as you've seen, it was so obvious from the UMAP that we actually knew that this would work well, because this is what J genes look like. If we can't do that, what can we do? So we expected this to work well, but then we put everything to the test, and we built the classifiers, and we quantified them. So these are the AUCs for public-private classification, and they work really well. Even a linear classifier does a really good job, which means that so this did better, but this did really well. The LDA did really well. So this means that actually there is a hyperplane in this hyperspace that has two sides to it, with public sequences on one side and private sequences on the other. And J gene classification, as expected, worked really well. And also mate cell classification worked really well. Shouldn't say really well, worked well if we were using alpha, which is kind of obvious because that's also the definition of mate cells, their alpha usage. But there is no J gene knowledge here. Just remember, these are only CDR3s. So it worked well for alpha, it worked well for beta, those not as well, and it worked well in the single cell representation. Now, when we when we sent this for publication, the reviewers, and you know the cliche, I want to thank the reviewers. Actually, in this case, I really want to do thank the reviewers because they came up with a lot of good stuff that is now in the paper and made it much, much stronger. One of the things was this thing. They asked, what would happen if instead of using your own transformer, you would use one of the available ones? And so there was TCR, actually, when I think even when the TCR BERT was already there, and TCR BERT is, I think, focused more on not on this, but on TCR peptide matching. But it's still it's a transformer that has been trained on TCR. So the reviewers asked, what would happen if you use the TCR BERT to do those same things? And you can see, for us, this looks really cool because we actually see the public and private, and it's really cool because you really see the J genes clustering going very well. So they asked, what would happen if you were to use TCR BERT, and what would happen if you were to use ESM, which is a general protein model? So we did those two things. So this yellow thing is kind of what TCR BERT looks like when you try to do this task of public-private, and you do see something. You do see public sequences somewhere along the edges, but I think most of them are actually hidden. So not as well as a CBC. And when you look at J genes, I don't think you see anything when you're using TCR BERT. And the same thing for ESM. You do see public sequences, which is an interesting phenomenon that we didn't go after along the edges, but you definitely lose the J gene clustering. So that was nice to see that this was working well. This is not the last slide, but I just wanted to mention this. For all this work, we need to thank Oracle because we used Google Cloud Computing and we used AWS, but Oracle, we need to thank them because they actually funded this well, not all of this, but funded the cloud computing for a lot of this by themselves. So I just wanted to thank them. And before I finish, I wanna tell you just in a few minutes what it is that I said that we were mostly working on. And this is the bulk of the work in the lab is on this is on the repertoire is a biomarker. And most of the students work on using repertoire, blood-based repertoire mostly to do tagging of samples. And so what we have done so far is we took a bunch of samples. And this is something Alona has been very efficient at, kind of amazing at being able to collect those samples. Those samples are extremely hard to collect. And working with tissue banks is difficult. The people in there are really, they're trying to help, but the tissue bank is not set up for this, especially not for blood samples and for PBMCs. So Alona was able to collect a lot of those samples. She probably collected more than 1,000 samples. Actually, I don't know the real number. I've been saying 1,000 samples long enough for me to be convinced by myself. But I think there are more than 1,000 samples that Alona has collected and processed into T-cell repertoires. And these are mostly samples from peripheral blood. And we have samples from ovarian cancer and breast cancer and colon cancers and more. And what most of the work is or has to do with is contrasting these samples of mostly cancer-associated samples with healthy tissue. Or I shouldn't say healthy tissue. I should say tissue from healthy individuals, because this is blood from healthy individuals, and seeing if we can tell those apart. You know, if a sample came from breast cancer or from a breast cancer patient or from a healthy individual. And so this is a paper that also was published just a few months ago. A paper, Miriam is the lead on this, and again, Alona collected all the samples, processed all the samples. So that paper discusses what we were able to do in ovarian cancer, because we were able to see um, the difference between repertoires that came from peripheral blood, um, that came from ovarian cancer patient, peripheral blood that came from healthy individuals. And that worked really well. And again, I'm sorry that this is a cliche and often a lie, but we should really thank the reviewers here, because they actually, I don't know if the reviewers might be here in the audience, but the reviewers for this paper actually noticed a few things that we never noticed and made this paper 10 times better by making it much more robust. Things like the specific subsampling that we used and things like that. So that is now something that we're very proud of, and most of the work again is around things like that. And that is the last slide. This is the lab, and so these are the great students in the lab. These photos were taken by Avital, and oh, this is, you can go to here and get to our webpage. And thank you.
Thank you for the very nice talk. I'm sure there are questions. Some people are even clapping. That's nice.
Oh,
Yeah. Yeah. I have a general or several general questions, but one question maybe, did you compare this? So you saw that public clones were on the edges of the clusters, right?
mhm.
So first question is, how much is that? Can this be? Could this be an artifact of UMAP plotting? And also, second question is, have you looked into whether there's any correlation with generation probability, whether these are highly probable sequences that, or basically what's the distribution of generation probability across the classes, so that maybe, and is that overlapping with the publicity? Yeah.
So your first question is, I don't know if UMAP, or I should say, I don't think that there would be an underlying reason for UMAP to do this spontaneously. But and that's what I said when I meant we wanted to quantify it, because UMAP is just the UMAP, and it's really cool to see those, to see the J genes cluster. It's kind of a phenomenon that you immediately see. But we wanted to quantify it and to see and to provide a metric associated with it. And that metric was the binary classification. So in a classification task, you're just as good as your representation is. If they were just scattered if the embedding just scattered those public sequences everywhere, you wouldn't have been able to find an algorithm that tells them apart. So from my point of view, the success of the classification shows us that the embedding actually holds those public sequences at their own special place that is decipherable from the features that are those 768 features per CDR3 sequence. You asked another question. Oh, the
About
generation
generation
probability.
Yeah.
Yeah, I don't know. We didn't go after this. I don't know that. I don't know. And to do that, we would need, to do this reverse engineering and to determine a V and J per sequence, because we don't look at the VJs,
Yeah.
but it is possible to get them, and we have them. But this whole thing happens without knowledge about V and J. But we could do that, and it would be interesting.
Yeah. Yeah. No, that sounds cool. So there's a question in the chat.
Oh, I don't
See
see
the
it.
question?
Just a second. Oh, now I see. If the UMAP embedding space is C groups and TCRs would have. Do you have any insight into this? What's it? I do not. I don't know why that is. I do not know what conversion recombination is such an enigma. I don't know what sets these TCRs apart. I do not know. Maybe someone in the audience has an idea.
Mm.
I don't.
Boy, you have a question?
Yeah, very nice talk. Um, so actually, it is related also maybe to convergent recombination. Did you try to build a language model based on the nucleotides as well? That's my first question. A related question is like, at the amino acid level, did you consider taking something different than a single amino acid as your basic unit? These are the topics.
Like what? You mean a representation of the amino acid, for example?
Well, what I had in mind is kind of we have a paper about the compression algorithm. So each one is, it has a different kind of. Sometimes when it's enough to get like one amino acid, that's okay. But then later in the sequence, you can get several of them, which will kind of be one token that go together. But even they're just taking two amino acids and try to build a language model out of it.
It's a good idea. We did not do that. And yes, we did try nucleotides. It didn't, for all the things that we were after, it didn't make a lot of difference, the nucleotide-based model.
You were still able to see the public sequences at the level of nucleotides the same way?
I think, I don't know, Romy was working on it for some time, kind of later. I actually don't remember all the details of what came out of it, but I do remember that we gave up on it because it didn't provide much benefit for what we were looking at. So I would need to ask her what exactly happened there. It could be interesting to use the other options that you came up with, like as in words, where you could use a lot of word embeddings. It would be interesting to look at a different set of embeddings out of this. Yes.
Okay. Well, can I ask another question, Victor? Yes, good. So, I mean, you advocated at the end of the use of repertoire biomarkers. I think there are like several papers out there, trying to advocate similar things. My question is like, how critical do you think it is like to have, in the context of T cells, CD4 and CD8 separated, but even more, how will like a single cell help in this classification? Yeah, because it's kind of weird that, all these papers just clump together CD4 and CD8 and get a, signal. But probably the mechanisms are different. So do you think it will matter a lot if people will sort it in advance?
Oh, you mean just actually the actual sorting before the PCR sequencing? I do think it will matter a lot. I do think it will matter a lot. Right now I shouldn't say there aren't that many, but we haven't been able to work with a lot of experiments in which CDR and any kind of sorting was done beforehand, and I do think it will make a large difference. Yes, we just didn't look into it so far. Also with single cell, yes, sorting and single cell. I expect that there will be many more of these soon, but so far we haven't been able to work with many of those. I do think that they will make a difference.
Okay. What about HLA? Did you sequence HLA as well in your 1,000 samples?
No, we did not. It's expensive as it is.
Do you regret
it? I regret nothing.
Okay, thank you.
Okay.
There's another question in the chat
Oh, in
again.
the chat. That was in compiling and curating a large data set. That is true. I should read it. A large part of the work was in compiling and curating a large data set, making the training of the models possible. Are you planning to share this training data set with the community? All the data is we didn't produce it. It's all public data. It's, for example, all the data that is, oh, you mean the curation, the actual curation. This is, I don't know if we're going to do that. Actually releasing such a data set out there is not trivial at all. You kind of have to maintain it, which is, and interacting with the people who use it, which is not something we have the funding for. It's not as if, if we release this data set and this doesn't work, and this, we don't have the manpower, the human power, the person power to handle this. So we could put it in the AIR data commons. It should already be there. These are data sets that have been out for some time. These are not data sets that we produced. So maybe a lot of it is already there. The single cell data, on the other hand, the single cell data is a thing in and of itself. Unlike those 2,400 samples that are for the bulk, the single cell data is something that we might be able to put together into one of the. There are a few single cell data depositories. Um, uh, uh, this is confusing. The questions. So the single cell data we actually might release as just a large data set. And someone here says that the adaptive immune code for the beta chain TCR is in the ADC. We mentioned the immune code at the beginning because that was just one try that Romy was doing, but it's actually not part of the paper. For the rest of the paper, we do use it on and off, but as you say, it's in the ADC. So that's good. Thank you, Felix.
There's a lot of chatter in the chat box.
Yeah.
But coming back to a question, oh, there's another question, but we go to this question after my question. So you found, as you said, core principles of TCR biology with your approach, but did we understand anything new about TCRs with your approach? And basically, maybe a second question, what does your approach add to the toolbox? Because we were able to differentiate between public and private clones, I guess, before, also with other approaches and things like that.
Yeah,
So
that's a really good question.
so how does it add? Yeah.
So adding to the TC, let me tell you how we use it, how we use CBC at the moment. So we find, and this is not something that I'm ready to talk about at the moment, but we find that CBC is a better representation of TCRs. When we try to do things with TCR, we don't use the TCRs themselves. We use their embedding, and that seems to be working very well. This is the reason I'm hesitant to talk about. This is working a bit too well at the moment. I'm worried that there's a mistake there. But when we use the embeddings instead of the TCR, instead of that representation, that amino acid representation is just what it's a concept, it's a narrative, and I don't know if it's definitely the best we could do at the moment. But as Gul said, for example, there are other ways to represent the TCR.
Yeah, exactly.
So that's one thing. We already use CBC as a tool in a lot of the things that we do. You had another question, another interesting question.
What does it add to understanding of TCR biology? So, or did you see any new TCR biology using your tool?
Okay, so I'm trying to say this in a delicate way. I'm trying to stay away from TCR biology when we work on this.
Mhm.
Everything I'm after is the utility, is the use. If I can make use of it, if I can do um, detection, cancer detection, if I can do those things, then this is what I'm after. I feel as if I'm not there regarding TCR biology. There is nothing intelligent at the moment that I could say about TCR biology. And not only that, I'm purposefully staying away from that debate because I'm all about the engineering in this case. I just want to make use of these amazing data and see if I can help in a more clinical way, or as clinical as it could get. So I think TCR biology would be, and probably is already being benefited from these models by people like you, who are actually after that understanding. But almost everything we do is utility based. It's not TCR biology based.
That's a nice answer. So, okay, we have two more questions in the chat. One, I will just read it. Would further pre-training on ESM on the TCR sequences you had before classification make any improvements? Also, are your trained models made?
The models are easily available. There are links in the paper. The I don't know if ESM would make any improvement. It's an interesting question because that's another way of providing embeddings. That's a good question. We should look into that.
Okay.
And there's another question here, right? Have you used the embeddings for some sort of binding classification tags? We did not. That is, for example, TCR BERT is doing that. There is another work at its very beginning in the lab, which is about binding classification and would probably make use of these embeddings at a later stage, but so far, no. I also hope that of these data will become available. So far, there is not that much data. So again, it would be really cool to look at this, but I feel as if we need more data.
Yeah, I agree. And then the last question, yes, so this work has been published in Science Advances, and there will be recording also of that on our YouTube channel. Yeah. So thanks, Sol, for a very nice talk.
Thank you.
Very nice questions, very
Yeah, that's really
knowledgeable
fun.
audience. Yes.